Harmonia axyridis. Each dot is a different population, and broadly speaking, what you can see is that the relationship shows that the higher the proportion infected, or the lower the number of males. So 0 is proportion males, 0 males. To the left-hand side of this graph, you can see that there's a relationship there where the higher the infection, the fewer males in the population, and vice versa. In terms of host behavior, there have been some lovely examples in butterfly species, so Crea species of butterflies, work done by Frank Jiggins, in populations where male killer prevalence had reached very great heights, so 90% perhaps plus of females being infected. And in these populations, instead of the normal situation where you have males lecking and females choosing, we have this sex role reversal situation where females lek and the males do the choosing. And very interestingly, there seems to be some evidence that males are able to select the females that are uninfected, which is a great example of natural selection trying to send the population sex ratio back in the right direction when it gets moved away from a one-to-one. Harmonia axyridis there are also experiments on Japanese Harmonia axyridis showing variable levels of female choice and indeed some male choice. And then the other interesting experiments have looked at the two-spot ladybird and one aspect of its mating behavior. So female ladybirds have a very strong rejection behavior of potential male mates, which can involve them going to extreme lengths to try and dislodge the male, shake off the male. They'll climb up plants, go on the undersides of leaves, and then drop off on their backs to try and dislodge the male from mating with them. But in female biased populations, that rejection behavior seems to be much reduced. And then the other aspect of reproductive biology that seems to be influenced involves the male sperm transfer. So male ladybirds will, I mentioned they mate for a long time. They'll mate for maybe up to 8 hours. And in that time, they'll transfer packages of sperm known as spermatophores, maybe up to 3 spermatophores in a particular single mating. But what some studies found is that in a population where there is, again, a female bias, so if we look at this graph, the St. Petersburg population, which is the purple bar in the middle, the number of spermatophores, the average number of spermatophores passed by the male reduces, as if the males can determine that there's no point in worrying about mate guarding or ensuring that they give the female as much sperm as possible because there isn't going to be another male along to compete with them. Worth pointing out that it's second male precedence for sperm precedence in ladybirds. So generally this prolonged mating may be an indication of some type of mate guarding, but that seems to be diminished when there aren't so many males around. Okay, so the final bit of reproductive biology I wanted to look at was this point that you might expect that when something has such a high dramatic impact on the reproductive biology of an organism, so killing off half your offspring, you might expect that the genome would fight back in some way, that the host would find some way of getting round this male killing phenomenon. And so we looked at a population of Chellomenes sedecimguttata, which is a Japanese polymorphic ladybird. So these two images are the same species. They look completely different, but they are, I promise, the same species. Small sample from Fuchu in Tokyo. And we did our normal thing of breeding these ladybirds and found 2 families where we had the low egg hatch rates and a female bias in the progeny, suggesting that male killing was happening in those families. We did some independent experiments where we treated the lines with tetracycline, and we could cure the females of that trait. And so they went on to produce more or less balanced offspring sex ratios. And we then did some work to analyze the 16S rDNA, and we identified a gamma protobacterium that was the causative agent. Interestingly, this one had very close similarity to secondary symbionts in aphids and in whiteflies, which are prey of the ladybird. So again, suggestive of horizontal transfer between the two organisms at some point in evolutionary past, but then something that's gone from being beneficial in the aphid to being an enemy, I guess, of the host in the ladybird. We then went on to look at trying to breed subsequent generations to demonstrate inheritance of the male killer, and we got some quite surprising results, where we found that so normally we would expect vertical transmission is not 100%. So if a female has 100 daughters, maybe only 99 of them will exhibit male killing, but it's, so it's pretty high, 99% ish, maybe down to 80% for some male killers. But we had around 50% of the daughters who appeared to be revertants, so in other words, had lost the male killing phenomenon. And we thought this was a bit odd, so we did some further breeding experiments. So this is just a phylogenetic tree to show the position of the male killer in the middle here of a clade which has got various different aphid symbionts surrounding it. So I digress. Back to the changing the sex ratio. So this graph illustrates what happened in these experiments. So what we did was we took individuals from this male killing line who had, when mated with their first male partner, produced either the blue line, no males, or round about a 50-50 sex ratio. And we swapped the males. And so what you can see is that fairly quickly, if we follow the blue line, that female started to produce male offspring. So the simple change of the male partner produced a situation where she started to produce round about a 50-50 sex ratio. For the other two lines, it's not quite so clear, but the red line shows quite nicely that sex ratio in her offspring dropped to a very low proportion of males. So this is suggestive of some kind of suppression of the male killing. And after a lot of breeding, our conclusion was that this was a single dominant autosomal gene that was causing this. The presence of the suppressor didn't stop the female from passing on the male killer. So the male offspring in those matings that survived, we were able to confirm via PCR, had inherited the male killer. So they were positive for the same bacterium that caused male killing in the other clutches, but the males were alive, so they weren't killed by it. So potentially that's working by killing the symbiont, and it's perfectly possible to get a positive PCR result from dead bacteria. Let's face it, we were just grinding them up and getting their DNA out. Or by reducing its vertical transmission, so perhaps stopping it getting into the germline cells, which would mean that it then doesn't get into the eggs and hence doesn't have that impact on the offspring, or reducing its replication. And any of those things might have allowed some bacteria to come through, but perhaps not enough to kill the males. Or there may have been some way, something more complicated, where somehow the bacteria couldn't identify it was in a male host, so the pathways that did the killing weren't turned on, or they were blocked in some way. So finding out how that works is a work in progress for some future project, I think. It's coupled to several unanswered questions in terms of how male killers actually kill, and also how they avoid being eliminated by host defenses. So ladybirds, like many other invertebrates, have antibacterial defenses, but somehow these don't eliminate the male killers. There's been quite a lot of research on this in particularly Drosophila and one or two other species, but nothing really to date on ladybirds. And so it'll be interesting to see whether we can actually generalize these results between different male killer hosts, bearing in mind that these different hosts, many of them have different sex determination systems. So ladybirds have a simple XY system, Drosophila have a dosage compensation system, butterflies have a WZ system. So can we generalize across these different systems? Can we generalize across the different male killers? Does the spiroplasma kill in the same way that a rickettsia does? We don't know the answers to these questions yet, so plenty more research to be carried out. What we have begun to look at is what other bacteria are found associated with ladybirds. So there's been a rapidly growing body of work looking at the microbiomes of all sorts of organisms, including large numbers of invertebrates. And Rafat Afan, who's a PhD student in the lab, has been looking at comparing Harmonia axyridis, which is a generalist ladybird, with the orange ladybird, which is a fungal feeder. So two ladybirds with quite different dietary lifestyles, and has found some quite interesting things, including that they share some core microbiota, but they also have some that are very distinctive and individual to those two different species and not found in the other species. He's still investigating the roles that these different bacteria behave, but some of them in other organisms at least have been reported to be responsible for impacting on the development of the host, or in creating resistance to insecticide for the host. So, you know, some very useful functions, as well as things that perhaps are more microbiota are more often better known for in terms of dietary benefits, and that kind of thing. So this image is just to show a kind of visual of when we have a ladybird genome sequence, and when the sequencing was done, all the reads were blasted to see what they were, and obviously the green ones indicated Coleoptera, which is what we were looking for, or other Diptera, which are the reddy pinky ones, or the purple ones, Lepidoptera. But there are also a large number of fungi and interior bacteriales in there, which suggests that, you know, the microbiomes of these things are enormous. And so that's a kind of ongoing project at the moment to try and identify what's in these different ladybirds, and indeed, you know, how different are they between different species. So with that, I'm going to stop talking about symbionts and move on to the other aspect of my research, which has been about the colour pattern polymorphism in the two-spot ladybird particularly. As I'm sure you're all aware, colour patterns have a variety of fantastic uses across different systems and species in ecology. Loads of different functions. Ladybirds, you can split them into broadly two types. So those ladybird species that are monomorphic, so where all individuals look exactly the same, and there are a large number of species that are like that, and that is perhaps the expectation we might have, given that these are meant to be warning colours. So one would expect that the individuals in a species would evolve to look like each other, so that any predator that learns they taste disgusting will avoid the rest of them. However, there are also some species that are incredibly polymorphic, and that goes against the expectation. So Harmonia axyridis, the harlequin, is one which has hundreds of colour patterns. Two-spot ladybird and its sibling species, the 10-spot ladybirds, are other examples where, again, in the two-spot in particular, something like 300 different pattern forms have been identified. We know there are various factors that maintain these polymorphisms. One key one in the two-spot ladybird seems to be that they actually do have a genetically controlled female choice. So some females have a preference for melanic males. It seems to be a single dominant gene, and those females will prefer to mate with a melanic male if there's a choice. Other factors that are relevant for melanic polymorphisms more generally, and certainly are true for some ladybirds, are these ideas of thermal melanism, where the more black you have, the quicker you can warm up, and so the quicker you get going early in the day, early in the season, and hence the more you can eat, the more mating you can do. And then things like industrial melanism, where camouflage might be an important element of developing those melanic colours. Mimicry is the final thing, and that maybe explains the two alternatives that there may be in ladybirds. So many ladybirds follow the phenotype of the 7-spot down at the bottom of the slide here, with the black spots on a red background type of phenotype. But the alternative that is found in a number of ladybird species is the red spots on a black background. And the two spots, as you can see above, actually one morph mimics the 7-spot type, the other morph the kidney spots or the pine ladybird or those red spots on a black background phenotype. So it's possible that both are maintained because they gain a benefit from looking like either of those rings. So we know that the colour pattern is controlled by reasonably simple genetics. It's probably a supergene with 3 kind of multi-allelic genes, but linked together very tightly. And then we know that there are likely to be some additional modifier genes which control dominance and some other pattern variations. So melanic morphs, I've got images here of the 3 most common melanic morphs in the 2-spot, with 2, 4, or 6 red spots on a black background. Typical form, which is the one people will be familiar with, has the 2 black spots on a red background. And then we have all these varieties in between, and you can see that the spots start to sort of spout little protuberances, and then they extend and they join up, and soon you end up with something that looks very similar to the 6-spotted melanic morph. So this whole range of intermediates is more or less a kind of continuous variety. So if we just look at the simple melanic 2-spot and typical 2-spot, this genetic cross just shows the simple segregation that you would expect. So a pure breeding melanic with a pure breeding typical, that's the red with the black spots, will give you an F1 where, because melanic is dominant, they're all melanic, and then an F2 where you have a 3-to-1 melanic to typical ratio. And so in my Daphne Jackson Fellowship, I took advantage of that simple breeding phenomenon to try and use RAD sequencing to map the region or the genes that were responsible for that colour pattern polymorphism. So I'm not going to go into how RAD works, although happy to talk more about it if people are interested. I think it's a fairly well-known and well-used technique these days. But essentially, what it gives you, rather than whole genome sequencing, which attempts to sequence the whole genome, RAD sequencing digests the DNA with a restriction enzyme and then sequences around the restriction digest sites. So what you get are representative sequences of the genome. But it's actually a very powerful technique. So we set up a cross using an F1 melanic heterozygote from an inbred lab line and a pure breeding 2-spots typical form to produce an F2 generation, which then now has a ratio of 1-to-1, so 1 melanic to 1 typical. 10-spot line was set up in a similar manner, except that the dominance is reversed in 10 spots. Just to make things slightly complicated, but I'm not really going to talk about the 10-spot work very much. So if you imagine that this represents a chromosome, with the blue bar being the locus for the colour pattern gene, and then the red bit being the melanic causing allele, what we can say is that a melanic individual will obviously have at least 1 copy of that melanic causing allele. It may be homozygous, so it may have 2 copies, or it may be heterozygous and have 1 copy of the red one and 1 copy of the blue one. Any typical individual will have 2 copies of the blue version. So when we look at our RAD markers, what we're looking for to find things that are linked to that melanic gene, that melanic allele rather, are things that occur in the melanic parent and all the melanic offspring, so the bottom line in this table, but none of the typical offspring and not in the typical parent. Okay, those are the ones we're looking for. The rest are interesting for something else, but not for this project. So we set up our crosses, we extracted DNA from parents and offspring, we made RAD libraries, we sequenced and assembled them, and we used a program called CryMap Improved to identify the markers linked to the locus we called phenotype and to create linkage maps. And from these linkage maps, which I've just kind of given you versions of here, one thing that emerged, which is quite interesting, is that the 10-spot markers seem to show, so these areas in blue blocks here, seem to show an inversion polymorphism. So the order of those markers in some individuals was clearly the other way around compared to some of the other individuals. And that seems to be a recurrent feature of ladybird genomes, that there seems to be a lot of inversion going on, and I'll pick up on that point again later. The circles are just around where the locus of interest is, and you can see that it's quite close in the 10-spot ladybird to this inversion polymorphism. So not long after we'd got those data back, two groups identified the gene Pannier as being the gene responsible for controlling the colour pattern variation in Harmonia axyridis. And they showed that this gene was highly expressed in the regions that where the melanic pigment is deposited, is produced and deposited. And they also showed that there were multiple inversions within the introns of the Pannier gene, which seemed to be responsible for the massive variations in colour pattern. And then the other thing they discovered was that Pannier was conserved in the 7-spot ladybird. So in at least one other species of ladybird, it seems to be the same gene with the same function controlling that deposition of melanin. So I mentioned we had a genome. We also had a transcriptome, both in rather too many pieces to be terribly useful, but information there nonetheless. And so another student in the lab did some work looking at trying to line up the Pannier sequence from Tribolium castaneum, which was the nearest beetle Pannier sequence that we had at the time. So when those papers came out, the sequences weren't available immediately for the Pannier locus from Harmonia. So she used the Tribolium Pannier sequence and our 2-spot RAD and transcriptome data, and was able to identify the exons as being conserved from Tribolium into the 2-spot. And subsequently, we've confirmed with Harmonia and with the 7-spot, showing these conserved exons, at least 5 of them. In some species, it looks like there may be more exons, but at least 5 exons are conserved in the 2-spot. However, the piecing together of the introns was not something that's gone so smoothly, and at the moment that's still a work in progress. And again, I can't help feeling that these multiple inversions will have confounded that issue and made it quite difficult to find something that we can align sequences to make sure they're in the right order. So one hope we have in our genomic investigation is to use longer sequencing reads. So this is quite difficult to see, but essentially in this figure at the top, what we have is a genomic read using MinION, so that's Oxford Nanopore data. So this is a 16 kilobase read, and then aligned to it, we have, it's a 16 kilobase genome read, and then aligned to it are MinION reads in the second panel and the bottom panel. And the MinION reads do vary in length. So essentially it feeds the DNA as it finds it through a nanopore. So if your DNA is good quality, you can get really long reads, and if it's slightly rubbish quality, then you get shorter reads. But essentially what we can see is that we've got reads that match more or less along this 16 kilobases. And then in the kind of the third panel down, we have reads from the transcriptome, and again, maybe slightly hard to see, but supposedly highlighted by the blue arrows. There are gaps in the gray. So the gray is the sequence, and the gaps indicate where the introns are. So one thing to notice is that the introns are reasonably small. So we're hoping that a sequencing combined with PCR approach may get us those intronic sequences. Okay, so with that, if I've still got time, she said, checking her watch. Yes, very briefly, I just wanted to touch on the utility of ladybirds and the Harmonia as an invasive ladybird, because something that people forever say to me and have been saying quite a lot this year is, oh, I found one of those awful Harmonia axyridis ladybirds, should I kill it? My response is no, never kill a ladybird, they're all wonderful. but it's worth, I think, just spending a couple of moments just to finish looking at the use of ladybirds as biocontrol agents. So they are massively successful in controlling things like aphids and coccids on a huge range of different crops. And of course, this reduces the need for chemical insecticides, so that's got to be good for the environment, good for other organisms. They are generally actually little affected by some target-specific pesticides, so there may be a kind of route for using biocontrol and pesticides, but they're definitely more cost-effective in terms of per dollar invested than pesticides. Evidence from the States, at least, would have us believe. Harmonia axyridis has been used for a long time now in North America, so introduced for biocontrol early in the 1900s, fully established in the 1980s for sure. It's been commercially available in Europe since the 1980s and is established in many countries across mainland Europe, I suspect all of it, including the UK, where it's now become notorious as this harmful invasive species. And it's absolutely true to say it's had a very detrimental effect on many of our native ladybird species and indeed other insects. In North America, in fruit production, it's reached pest status. And there's a terrible problem with something called ladybird taint, because the chemicals the ladybirds harbour taste disgusting, not just to things that might potentially prey on them, but also to human beings. And they can, for example, ruin your wine crop if they're not removed from the grapes before the grapes are crushed. So there were lots of kind of newspaper articles that went on about how awful ladybirds were. And this graph, it's a little bit out of date now, but it shows the spread over the years. So coming into East Anglia in the early 2000s and spreading across the UK to pretty much cover the entire UK inside 10 years. Is an incredibly fast invasion of, the fastest spreading invasive species known. So it will eat other individuals of its own species, as well as other species, including invertebrate immature stages of many things that we all love: butterflies, moths, beetles, other insects. And it tends to outcompete many of these things for a number of reasons. Primarily, its wide dietary range and the fact that it, will eat plenty. It also has a high reproductive ability, and whereas many ladybirds will have one or at most 2 generations in a year, the harlequin has been reported to have up to 3 generations in a year. So really big opportunities to multiply its numbers. So how can we control it? Questions for the audience. Could we use insecticides? I'm never in favour of insecticides if we can avoid them. Can we use pheromone trapping? Complicated, not sure it would work, expensive. Can we introduce enemies from its native range? Well, again, we all know how badly that can go wrong. We get rid of one pest and we end up with another different pest. Could we harness the male killer? Is the thought I will leave you all with. Is that a way of controlling it to a limit? So just a few people to acknowledge, particularly funding bodies and other collaborators over the years. And with that, I'll say thank you and take any questions.
Thank you very much, Tamsin. So now I'm happy to take questions from either the people at the University of York, or if you're watching on YouTube and you'd like to post a question in the chat, go for that, and those questions will be passed on to myself and Tamsin. I can start the question for now. So in terms of the bacteria or the ladybird, is this how often is these male killing bacteria, how often are they a dead end? Often do the ladybirds go
yeah. So in ladybirds, I'm not aware of populations where they've got to more than about 50% prevalence, and so that means there are still plenty of males around to do the mating. In some of these butterfly populations, they've obviously started to be a problem. So the reports indicated that many of the females were virgin. They were dying as virgins because they couldn't find a male. And so in those instances, you would expect that population will just die out. The alternative, which interestingly one can speculate at least has happened in the past, is that the thing evolves to be to do without the males. So parthenogenesis, and there are some nice stories of parthenogenetic wasps which don't normally produce males, but if you feed them antibiotics, they produce males. So so they still retain the ability to produce male offspring if you get rid of the bacteria, which is quite a nice story, I think.
So there can be multiple ways out.
Yeah.
Hi, Tamsin.
Hi, Kanchan, how are you?
All right, how are you doing? I was just going to ask something on the same topic. I mean, it was interesting. You showed that there was, it was really fascinating talk. We talked about a wide range of things. One of the things you said, there was a lot of variation between populations in the proportion of females which harbor the male killing, and you were kind of speculating as to there were multiple reasons why there could be variation. And have people actually investigated that, the factors which correlate? I mean, I don't know, the availability of food.
Sorry.
So within a species, what factors are affecting the proportion of infected females?
I think there are certainly things we've discovered along the way. I'm not aware of studies that have set out specifically to address that question, but different male killers have different vertical transmission efficiencies, is one thing. And so, so for example, rickettsial male killers have only about an 80% transmission efficiency. So, as we're all thinking about the way things transmit with COVID at the moment, I think, the idea that you have 0 males in your clutch if you've got a spiroplasma male killer, for example, or a Wolbachia male killer, but you still have 20%. If you have a rickettsia, for example, might very easily make a difference. There are other things like the clutch sizes will make a difference. So some species lay smaller clutches. Harmonia lays pretty large clutches of eggs. Some, so they maybe have, I don't know, 25, 50 eggs in a clutch, whereas other species maybe only have, 4 to 8 eggs. So again, that makes a difference just in terms of the rapidity, I guess, of one infected individual being able to spread.
Do you get multiple infections by any chance?
Well, that's something that I'm hoping we'll identify. Not within a single individual, as far as we know. Anecdotal work with other organisms would suggest you can get, so in spiders, we've seen instances where individual spiders have got rickettsia and spiroplasma, or Wolbachia and spiroplasma, for example. But they're not male killing in spiders. Yeah. They're doing something else.
It seems it must be fairly rare in the ladybirds. You would have caught it when you're doing it. Yeah.
I mean, what's interesting to me is that when we did this work, we were very naive about wider microbiota, and our PCRs, fairly straightforwardly, I did clone and sequence my PCRs rather than just doing the PCR and then sequencing the product. But even so, the vast majority of clones were the male killer in each case. Whereas now, if we do a 16S PCR, it seems that we can clone. So Rafat's work, he's cloned all sorts of different things. Almost every clone was a different bacterium. So I think when there's a male killer, it obviously really dominates that microbiome. So it is the majority of the product that you're getting, and hence the majority of the sequence.
The other question I had was with regards to this. You were saying you had an spots in the 10 spot. He said there was an inversion polymorphism within the 10 spot. Did you find anything in the 2 spot? Why do you consider it to be a superhelium spot?
We haven't really investigated that any further. So in the 2 spots, so our RAD analysis so far in the 2 spot has simply been things that were linked to that phenotype locus, and in the closest linked markers there was no evidence of an inversion. But of course, bear in mind that this is all one family. So the individuals in the thing were from one individual pair. So, you know, it's quite possible that if I'd picked a different individual, I'd have found something different. And we haven't yet looked at a broader population-based question on that.
Sounds really interesting and very complicated with all these.
Keep me going for decades, I think. Thank you.
Yeah, thank you.
Could I ask about the inversion polymorphism? Tamsin, do you, in the region where you think the gene is for the colour patterns, do you see less recombination?
So interestingly, one idea that was propounded a long time ago, and I think still potentially holds true, is that those different colour pattern variants in the 2 spot are due to recombinations within that colour pattern locus. So the locus that I said has probably 3 component genes, but tightly linked. The suggestion is that recombination within that locus generates some of those intermediate variants.
I see.
Whereas there's allelic variation that generates the melanic versus typical, if that makes sense. So there's a kind of big switch, or potentially there are little switches. So I would suspect, you know, it's for me, it's really interesting to see the work showing the inversions in the Harlequin Pannier locus. Yeah.
Does sound complicated at the moment.
Long reads, I think, are the way to go.
Absolutely, yes, certainly. So we're now at 6 minutes to 1. So I propose that unless we have more questions, that we round this one up, given that Tamsin has somewhere to be.
I'm happy if you get more questions later on. I'm happy to answer questions by email. That's not a problem.
So I'd like to thank everybody for coming, and everybody on YouTube. And we'll plan to do this again on YouTube at some time, and we'll send out tweets when we do this again. And thank you very much, Tamsin, for a wonderful talk.
Thank you very much, and good luck with the rest of your